The Innovations in Soil Science presentations are funded by the Australian Government's National Landcare program and was delivered by Soil Science Australia, Agriculture Victoria and Gecko Clan Landcare Network. The following presentation is introduced by Brad Costin from Agriculture Victoria.
Introduce Gary Clarke, the Trobe Uni's Gary Clarke up for the next talk on subsoils, understanding what's below the surface.
Okay, so just to draw you, if you're not aware of the Australian soil classification, so how does it work? This is what they call just a quick key to the Australian soil classification. There's a longer key which we use in depth if we're going like tomorrow to describe a soil profile and we want to identify the soil type. But this one's a quick key, and I just want to highlight the texture contrast soils. Most of you are aware of texture contrast soils. They're very common in our part of the world. So these are I'll just go back. So there's three types: curasols, they're acidic in the subsoil; sodasols are sodic in the subsoil; and chromosols tend not to be sodic or acidic in the subsoil. So firstly, the distribution of chromosols in Australia, and as you see, very common through New South Wales, Victoria, high rainfall areas. Then we look at the sodasols, and once we put them together, we know we've got a lot of texture contrast soils in eastern Victoria, and eastern New South Wales and southern New South Wales. I haven't put the curasols. Curasols not quite so common, but they are, as Helen's data was showing, ferrosols, curasols, and forgot what the other one was, all close together in Tasmania. Right, so what's a texture contrast soil look like? So typically, they don't come out, it's a bit dark for you guys, but look, we've got a loam surface soil. They could be sandy loam, silty loam. Probably this part of the world, they're probably more sandy loams over a high clay content subsoil. And with this is particular sodasol up at Benalla, and so the subsoil is sodic. So what's the matter with, or what's the constraint? Think about constraints and whether it's going to impose something on your production. They're not, this talk covers a bit more probably cropping than pasture production, but I'll get onto a bit of pasture production. Okay, so the high clay, dense clay subsoil imposes a couple of constraints. There's a large amount of very small pores. So that pore distribution is important because if we've got lots of small pores, there's a lot of water that's just not available for plants. It's just in micropores. And so there's a large amount of unavailable water and a low percentage of plant available water. Usually there is a very narrow window between the field capacity and wilting point in a lot of these high clay content subsoils. And lastly, for crop production and look, even pasture, particularly something like ryegrass will be restricted to that surface soil, and there's very few roots down into the subsoil. And particularly, and you would have noticed like Jason's little picture of taking on that little machine onto the paddock. Last year particularly would have been a lot of waterlogging. So, and sometimes the surface soil can be fine, but you'll notice in that subsurface there'll be quite a lot of, it'll be very wet, particularly like last year, and that's when you lose your machine, get your machine bogged. And so, and getting that water in and out, and I'll talk a bit more about that, is always problematic. It takes a lot of time for that water to infiltrate and fill that subsoil. So normally, in that sort of constraint, roots just give up the ghost. So an example of this, last year was so wet and we held a soil judging up at Ballarat. So all the soil profiles had lots of water, but a common observation remember the last 2 years have been really wet, some of those subsoils are quite dry. Why is this so? Why is the subsoil dry and it's super saturated at the top? So what's happening here, it's very hard to see from this picture. First thing, a lot of organic matters are up the surface. Sometimes this is quite a deep topsoil on the side up at Ballarat, Federation Uni. But sometimes those topsoil or organic layers only about 0 to 5, no, not even 0 to 10. So then below that, we've got, you see that little, it doesn't come up on that photo, I can see it on the screen, but you'll have a bleach layer. We call that an A2, and see these little bits here? That's that, once we cut the pit, the A2 is spewing out onto that subsoil. So this is what some people term spewy subsoils, or subsurface really, because that subsoil is properly right down here. So that A2 just becomes super saturated, but that water's not penetrating into that subsurface, into that subsoil. So that's something that you need to keep in mind. And they tend to be highly dispersive. Like, we'll show you this tomorrow, the Emerson dispersion test. So surface soil, there's a little bit of dispersion happening here that was probably the lower A, probably the A1. But once we're getting into that subsoil, it's quite dispersive. Now, one thing I need to make you aware of, when you're getting soil tests, you get cation exchange capacity, yes? But you know what that ESP value is designating? It's been a lot of research showing that about ESP of 6, we've got a sodic subsoil. But sodicity doesn't always mean it dispersed. Usually does. Sometimes you get this dispersion happening and it's not sodic. So you've got to be, the chemical measure is a little bit imprecise at times. Doesn't tell you it'll disperse. So you need to do this test, because you can get a non-sodic surface soil that can look like, probably not quite as bad as that, but it can disperse, and that could be due to the low EC, low amount of carbon, number of reasons. So that's something I'd like you to take home. Just do that test, and as well as get the CEC done. With pasture production, it's okay, you might think about deep ripping, you might think about different pasture species. It's a lot more challenging because you're not getting that, there's not the potential there to increase your yield like with grain cropping. So I started off in doing work in my masters and with University of Melbourne, and we looked at deep rip and gypsum, a couple of sites. And so that was back in 2004, finished my masters, and it had a limited effect. And it's because gypsum is really fixing up the dispersion. So think, if it's dispersive, it will be gypsum. It may be gypsum responsive, but it depends on that CEC. That's when you need the CEC, if it's got a high amount of exchangeable sodium, you can put that calcium in there, that calcium sulfate, the gypsum, and it'll lower that CEC, lower the ESP, sorry. So, but with it's just doing one thing at a time. So this is where I got onto the subsoil manuring with Peter Sale and Professor Tang's group. So the idea is that you're trying to get a number of processes happening at once. Okay, so I've got some of the results. It's been shown over a number of years. You can see this data tables from 2005, 2011, and the average was 57% increase in yield. So why was this effective? So I'll go through the process. Okay, so an implement on a tractor, and I've done things like one tine and a bucket and throwing bucket down a chute to bigger machines that have been developed to do this. So like for instance, Terra Nova Ag have got a big machine. Other machines, Elmore Compost people, they've just developed a machine. So the idea is you're deep ripping and you're some way of getting down a organic manure, particularly chicken manure, sometimes compost. Composts are coming into play a bit, and we found, and it can be the organic amendment plus some nutrients added. That's the important part. So importantly, because of the multi-mode of action and what the consequences and the results, the most important part was increasing the water use efficiency. Or was it? So we're terming it rainfall use efficiency. So it's important for capturing rainfall rather than making the plant more efficient use of the water that's in the profile. We've found that it's by opening up that subsoil and getting that infiltration into that subsoil, you can capture rainfall when it's maybe not optimal. When's the non-optimal time? Maybe late summer, autumn. And when even for pasture or crop production, the biggest growth time's that late winter through that spring period. When it gets warmer, everything's going gangbusters, so you need that available water. So hang on to that thought about that available water. So whether subsoils are not physically constrained and you've got an adequate amount of rainfall, and there was no toxicities in the topsoil or subsoil. I'll cover a little bit of that in a minute. Okay, so in rain-fed crops, there's been a lot of evidence, and like I showed you that table, that the process seems to work. But we've tried looking at it in rain-fed pastures, but there's been no real work to try the process in rain-fed pastures. No one wants to spend huge amount of money on that manure in the process. Okay, so, but the evidence that it does transform that subsoil structure, and the plants do use more subsoil water, and it does capture more water, more rainfall, I should say, and it stores more rainfall in the subsoil. So it's important to capture it, and then it's got to be available. So that means you've got to have those roots going down into that subsoil. So it's no use getting that water into that subsoil, and then the roots can't use it. And it does reduce waterlogging. That's going to make more efficient plant growth. So it ends up using soil water much more effectively. So the transformation process depends on a lot of things happening at once. So most important is that manure, and we've found you've got to have optimum nutrients, because no use using manures that are low in nitrogen and phosphate. They're the two most important. And then you get lots of stuff happening at once. The manure will kick off that, or the nitrogen in the manure will kick off the root growth, and then you get associated microbial activity, exudates, which help bond those clay particles into aggregates. And so it's a combination of root growth, microbial growth, all those things happening at once. Well, again, as I said, the nutrients are important. And why we're adding a little bit of nutrient to the compost, because if you have a lot of growth and there's not sufficient nitrogen, you're impeding that growth. So that's important, but you're relying on mineralisation of the nitrogen from the compost. So just an insurance policy, you need to add some nitrogen to that, to the compost or the chicken manure, depending on the nitrogen level. A lot of chicken manures can be a bit low in that nitrogen level. So, so nutrients will stimulate root growth, fungal hyphae, and another important aspect is that dry re-wet. The aggregation is that biological, physical and chemical processes all working together. So a lot of subsoils are wet. Once you're getting that root growth, it's pulling out that water and transpiring it up through the leaves. So that helps dry the profile out, and you're getting that shrink swell, which helps form aggregates. And once you're getting aggregates forming, you're getting better infiltration and the increased bucket size again. Okay, so just a couple of things to show you why, just evidence to show you how there was more subsoil water. So here's our profile again. We did some subsoil manuring and booming crop in 2005. Why this particular slide, again, you can just see a little bit of green in there. Just imagine you can see some flag leaves that are remaining green. Suddenly, in 2005, we had sufficient rainfall during winter, sort of early spring. It started to dry off again. So there was water stress. So the plants that were on top of the subsoil manuring, they started accessing some of that subsoil water. And you can see the yield increase there. Okay, so what we did was measure how we captured more rainfall. So we had, over summer, autumn, there was a couple of 100 millimetres, and the commercial paddock was 75 millimetres. So, and whereas subsoil manure was 150 millimetres. So double the amount of water that was infiltrated into that subsoil. So you would have everyone's seen texture contrast soil. You get that if you've got 200 millimetres of rainfall over a fairly short period, you would get a fair bit of waterlogging. So and a lot of that will either, why they came up with raised beds in southwest Victoria's, because of that waterlogging problem. So that water is always shed off in a raised bed situation. Okay, so, and again Peter and Gecko measured back in 2012 at Stewarden, had subsoil manure plots, control plots, and they measured, using rain-out shelters, measured how much was getting. They wanted to measure the crop lower limit, so this is not a laboratory measurement. You measure it so there's no more water being accessed out of the subsoil, and then you get the drained upper limit, which is your field capacity. So that's important. Between saturation and field capacity is really how much aeration is in that soil. So what we had, how much water did we store? So topsoil, fairly similar, which is subsoil manuring doesn't alter the physical properties of those surface soils. A lot of the surface soils are fine. You would have seen pastures on texture contrast soils. You go down to, if they've been managed well, 10 centimetres, they're probably fine. They're nice root growth, etc. But in the subsoil, with the subsoil manuring, there was, you could see more than double the amount of stored water. So there was an extra 78 millimetres of plant available water in the subsoil, which is enough for an extra 3 tonnes of wheat grain per hectare. Okay, so could we reduce waterlogging? And this is something this is why it's interesting. We haven't really measured this, but sometimes anecdotal observations are good. Like if you're talking to farmers, you pick up some of these anecdotal observations. They were talking before Jason was, no, grid sampling's too, just use observations. And look, you need a little bit of both. I reckon the farmer can observe something. If you can measure that bit of grid sampling or whatever and follow that up, that can be useful. Okay, so you did the treatment there, you've got a baseline. And this is what I'm finding, you need that baseline in if the farmer wants to innovate, help him innovate, but tell him he needs to do some baseline measurements. Because there's a lot of anecdotal information out there in the public space. But one thing we're looking at there's another interaction as well as aggregation in those soils. With these organic amendments, we've been doing some more work at La Trobe, thinking about that, or measuring the rate of transpiration, respiration of the plant. So the plant's got to take in CO2 and build carbon, but they're transpiring water at the same time. So, yeah, okay, so what's this matter? So if we can study those two systems, can we make an efficient use of the water and build more carbon? So we've been, Joanne Wang did some column experiments in our glasshouse at AgriBio, and so in this column here, what we're doing is growing some wheat in a smaller core. The reason why we're doing this in a smaller tube is to make sure those roots just enter that subsoil. So this was filled mostly with topsoil, and then in here, here's our hostile subsoil. So we've dug it up from a site, crushed it, and then put the amendment. So we've got an amendment band like in our subsoil manuring, and we've put all these moisture probes, and we're watering. Importantly, we're watering from down in that subsoil, because what we find with these setups if we have this set up in the glass house, when we water from the top, it just tends to use water up from the surface soil. So interestingly, we had a deep fertiliser. This is some of the speculation about subsoil manuring. It's just driven by nitrogen. Okay, so we've done some of that work to try to counter. So this deep fertiliser NPKS, and so shoot growth 15.4 grams, water transpired 4.8, transpiration efficiency 3.19. So once we put deep nutrient-rich organics, we got more shoot growth, same amount of water transpired, but you can see we've much improved that transpiration efficiency. So that's going to be, that's that amount of biomass we're producing for each millimetre of water that's transpired. So that's something interesting that might help in the future. Okay, so what, and there's further work to be done, but there seems to be interaction between the amendments, the roots, and there's signalling there, there's hormones being released and they're regulating those growth hormones. So this is what's turning on and off that stomata. So normally it's quite a linear plant growth, you need more water transpired. So if we could alter that, so we bend that curve a bit, so we're still getting the same amount of biomass, but with less water transpired, that'd be wonderful. All right, so thinking about, I thought, what more can what's the next step after subsoil manuring? And so Peter and I have been thinking about, well, can we use cover crops? There's a lot of action in the regenerative farming. So where are we going to from here? Could cover crops, multi-species cover crops help with texture contrast soils? So if you think about that, going back to the model of the subsoil manuring, we're adding nutrients to stimulate lots of root growth. With multi-species cover crops, you're planting a lot of things and you're getting a large amount of growth if you've got either the soil water or the water falling on that crop, and you're getting dense root growth. And this is the advantage of a multi-species cover crop. There's a range of species there. They've got different root systems, and you're putting a lot of carbon in the soil, probably improving you're driving that soil biology to improve that soil structure, you hope. There's been mostly anecdotal evidence saying, well, let's do tillage radish, and this is applicable for you guys. You drain pasture, particularly dairy, because dairy's problematic. It's always in a higher rainfall area, gets waterlogged in winter. How can we drain that soil? We could subsoil manure, but if we just want to sow something within that pasture, but it's a bit like buyer beware with tillage radish. I think you might get an effect by just drying out the profile, but is there a long-term effect? And I've got doubts on that, because you can get possible bypass flow. Has anyone heard of bypass flow? What's that? So bypass flow has been shown if you grow stuff like lucerne, woody plants, they put down very large root systems, not like a cereal or a pasture where they have a big root volume. With those other, as I said, lucerne or tillage radish or a woody perennial, they put down a lot of large roots, and there is evidence to show, we showed it at Benalla. One paddock had lucerne, one paddock didn't. The one with lucerne had bypass flow. So we got the same amount of rainfall and it went in right down below the root zone. So that's the thing with, lucerne is promoted for amending the subsoil, but it's forming root channels, yes, but you've got to be aware in this, and we're going to get more of these high rainfall systems. They're just inevitable with climate change. You could get a lot of that bypass flow. Doesn't mean to say you could use lucerne and then follow it up with something else. That could be another possibility, but yeah, there's evidence to show that I would think that tillage radish, it'll be a short-term effect unless you're doing something to keep that root growth going. And that, I think that's why there was a longevity effect of subsoil manuring, because you put down a lot of organic amendment deep into the soil, and that mineralised over 2 or 3 years, especially when the year was a little bit dry. Okay, so that's the... Okay, so my next, thinking about other soils in other regions. So this is a Curacao down in West Gippsland. High rainfall area. Curacao is still texture contrast, but it's acidic in that subsoil. And these are very acidic. And it's not just below Jason's pH 5, or it's got lots of aluminium as well. So we visited the property where some people might have heard the Soilkee pasture renovation method. So they're using it's a pasture it's renovating within an existing pasture. You don't need to change your pasture with cover crops, and they're doing it in strips. It's quite clever. So multi-species cover crop, but importantly, there's a couple of processes. You're not just putting down that multi-species. It's cutting the chaff that's there, and it's cultivating where the seed's growing. So it's only sowing in strips, and it's cultivating that soil. So you're getting a nice area for seed. As long as you've got moisture, you've got seed germination, and those roots will take off. And importantly, the way this process has taken time to build soil carbon and soil structure, it actually is real, that change in soil structure, but it's twice a year sowing up to about 60 kilos of seed per hectare. So quite a lot of seed sown twice a year. And importantly, grazing, and this is cell grazing. So probably unlike the guys up here, paddocks are much smaller. You can really graze if you've got that rainfall, which they have. You've got that pasture production. You can crash graze those paddocks, and you can easily put them in there for a few days with a electric tape, and they eat so much, and the rest is just trampled into the soil. So those two, remember I said about chaff and grazing, are really key to that process. And think about the input from the animal, the dung and urine. So that's the machine being pulled behind a tractor. So it's hard to see in that photo, but little cultivators at the front of you couldn't see the front of the tractor. They've got chaff cutters, so they're taking it off down to the ground and sowing seed at the same time. Okay, so we Peter and I went to the farm. We sampled 2 paddocks. One was FF was the family farm. Should have made the font a bit bigger, And the orange bars were the Soilkee, because there was a claim that the Soilkee was amending the soil pH. Quite important, Jason. He's bumped that soil pH up immensely. Right, you can see how acidic that parent soil is. This was water pH too, so pH of around 5 right down the profile. And with Soilkee, all alkaline pHs. And okay, so what's the difference? They were different adjacent farms. They weren't next to each other, the paddocks, but the family farm had really low input. And sadly, I didn't take photos of the cores we pulled out, but the root growth was no deeper than 5 centimetres. Really, really poor. So acidic, and if you hadn't limed or anything, it's just struggling. So not improved pasture, no lime. But on Soilkee paddocks, they've been used for dairy, had lots of lime, 5 tonnes of lime over a couple of periods. So, yeah, so I'm a little bit sceptical of the alkalisation. We've had lots of discussions. You can't alkalise just by putting in plants. You need to put that magic thing called lime. Okay, so the soil structure improvement is really quite incredible. You can see why they got carbon credits. So you can see these are not my photos, but this was like a control paddock. I think they cultivated. Oh, is it? No, that's. Oh, and middle one is not amended. Then that's the Soilkee on the right. So these are the take home. You need 1,000 millimetres of rainfall. It was a highly acidic soil profile. So there was a soil carbon increase, but a lot of it's due to the high rainfall, high input, biomass twice a year, and the seed input, cell grazing, dung and urine. And I should add to that, and then the lime as well. So Helen would agree with me, probably Jason. You're probably not going to get the biology happening as happy, or it's not going to be as happy until you get that pH out of that acidic region. This is my emphasis. If you do something, you need a baseline. If you do a treatment, you need a plus or minus treatment, even if you're doing strips. So this guy he didn't have the starting before he did Soilkee. He had soil tests, you know, farmers, they're in the filing cabinet somewhere, and it was acidic, but he put on 5 tonnes of lime. So good on him, he's come up with the method, but it's just, it'll work, but it takes a lot to get that soil carbon, you need you need that biomass input. And it's like Jim was showing, you need the nutrients. So by putting in that seed, dung and urine, producing biomass and putting it back in. Okay, so Matt Marnie, he had some funding. He's an agronomist in the Yarra Valley region. He's funding from MLA to look at subsoil constraints, poorly drained soils. So he mentioned 1,000 millimetres of rain again. So normal winters, a lot of farmers have got paddocks that are poorly drained, they pug, and again, they're not, some of those farms, fairly small paddocks, they can't, because one of those, at Coldstream, he's got lots of different parts of his business, can't move the cows off, so they're on there pugging away. So we want to drain, so you think, well, poor drainage, subsoil constraints, let's do subsoil manuring. So this is what you need to investigate the subsoil. So as you can see, yes, very mottled subsoil, poor drainage. Okay, so we want to improve drainage, but we analysed the soil. So this was at one cold stream. It was quite acidic. Exchangeable aluminium's fairly low. Next place, burolite. Lo and behold, pretty acidic, but look at that aluminium. So what are you going to fix? It's a bit like what Jim asked before. What are you going to fix? Drainage or the pH? So this is what we're trying out. So amend the acidity priority 1, subsoil improvement would be priority 2, and then the solution with the trial. We're going to, it's been deep ripped in strips, deep rip plus lime, deep rip plus chicken manure, and then we're going to Soilkee across those strips. So by doing that, we can just do some paired sampling, and we could do some basic t-tests or basic statistics. So what is my... And also we can do biomass sampling, soil core sampling, and as I said, paired sampling. So take home, try to innovate and talk to farmers, talk to agronomists, talk to soil scientists, talk to extension, and let's work together and try to do things in a methodical way and get a bit of data so we can, back up what we're doing and move ahead. All right, thank you.